Session, Transforming Future of Microbiology, Perspective of Next Generation. This session will be moderated by Dr. Sushma Pednekar. Dr. Sushma Pednekar is an associate professor at BJ Government Medical College and Sassoon General Hospitals, Pune. With 14 years of expertise in diagnostic microbiology and 10 years of focused work in hospital infection control. She has extensive experience in nosocomial infection surveillance, antibiotic policy development, and infection control training for healthcare workers. She has managed NABL accredited laboratory operations and led COVID-19 diagnostic efforts, including RT-PCR and Gen expert testing, as well as investigations into COVID-associated mucormycosis. Dr. Pednekar has 12 national and international publications in a recognized journals, and she has a postgraduate guide also. She has received multiple awards, including MedWorld Asia International Excellence Award in Medical Education. Additionally, she has conducted state-level training programs in infection control and COVID-19 preparedness. She is also in charge of scientific committee of this conference. I will hand over mic to Dr. Sushma Pednekar to start the session.
पूर्णपणे बरं का प्रश्न मला थोडं पण कमी जास्त नसेल एवढं आता तिकडे बोलताना तुला सांगितलं ना बोलताना इकडे जायचं
Thank you, Dr. Vaishali. Generations are changing fast with technology in today's world. It will be appropriate that we give new generations a chance to express their ideas, and this will be an opportunity for us to understand their perspectives. With this, I call upon our first speaker, Dr. Akshay Karyakarte. He is a clinical microbiologist with close to 8 years of experience holding an MD DMP in microbiology. He currently serves as assistant professor at St. GS Medical College at KM Hospital, Mumbai, as a supervisor in the clinical bacteriology laboratory. Additionally, he is actively involved in infection prevention and control activities, undergraduate, postgraduate teaching, and coordination of interdisciplinary clinical services. With a keen eye for research, he has numerous peer-reviewed articles in international journals and many more in pipeline. He also serves as a certified peer reviewer for esteemed publications like the British Medical Journals, among many others. Always on the look out for scientific improvement, he is constantly dabbling in cutting-edge technologies, whether it be getting certified in CRISPR, gas gene editing applications from Harvard University, or being a key supervisor for the multi-top MS machine installed at the Department of Microbiology at KM Hospital, Mumbai. With this, I am pleased to welcome him to talk about his perspective about multi-top MS technology and its future application. Dr. Akshay Karyakarte.
Good morning to everyone. First of all, thank you, ma'am, for that very kind introduction. This conference has been an intellectual feast, and I'm truly honored to be standing here and talking upon something which is very close to my heart, that is multi-top MS. So, as regards to perspectives of the next generation and what lies ahead, I would like to first begin with what actually MultiTOF is. Everybody knows that, but so MultiTOF is an abbreviation which is matrix assisted laser desorption ionization time of flight mass spectrometry. So what it does is it is an automated identification system, giving accurate identification for bacteria, fungi, microbacteria in minutes. So the reduction in time in identification of microorganisms and pathogenic organisms is the key why multi-top MS has been so popular in the recent times. So how it works? So how it works is actually hidden in its name. So matrix assisted. Matrix assisted means the matrix which we are putting upon the microorganism, the pure culture of the microorganism. It converts it into biomolecules which are then inserted into the machine, and a vacuum is generated over there. And in the presence of a vacuum, a laser is bombarded upon that, which makes those particles or those biomolecules ionized. So matrix associated ionization has been done. And addition of an electric field from negative to positive charge makes it helps it to dissolve from the slide which has been. So matrix assisted laser desorption ionization. Now the addition of an electric field makes these molecules, makes these proteins which are inherently negatively charged, rise up from the slide up to the detector. So this is now, of course, the time which is required for the particle or the protein to reach the sensor is defined as a time of flight. This is going to depend upon how much charge it holds and how much mass it has. So this time of flight is a function of mass by charge. So m upon z is the important function over here. And these particles which are reaching the sensor are then read and analyzed by mass spectrometry. So all these together come together and help us identify the proteins, identify the biomolecules which are inside the microorganism. So we can see how it works over here. Right, so how does it identify them? Just breaking the microorganism down into its constituent biomolecules is not sufficient. So what happens is this mass spectrometric analysis produces a spectrum. This spectrum has unique peaks which correspond to the specific mass upon charge ratio. And this is then fed into a database which is specific to those specific vendors which produce that machine. So what mass spectrometric analysis has been obtained by the microorganisms which are being processed on the data, they are going to be compared with this database. And if these peaks match with them, it is going to be identified as so and so organism. So this is how it works. So what has happened in all this is two important things. First important thing is that the time required for identification of these organisms has reduced drastically from hours to minutes. But also most importantly, we require a pure culture for identification of organisms in biodegradability. So the next thing is, so what? What if we are able to identify them in minutes? What happens then? Because if it requires a pure culture, how much time is actually being saved? Because overnight incubation and inoculation of samples, all those processes are not being bypassed. So in effect, what is helped? How is the patient being helped for that? So the answer to that is what is being done right now. What, other than the identification of these pathogenic organisms, what is multi-top MS technology being used for? So it is being used for detection of viral pathogens. Most importantly, in the pandemic, it was used very frequently and very rampantly for the detection of SARS-CoV-2 in a qualitative way, not a quantitative way. Then identification directly from positive blood cultures. This, according to me, is the most significant advancement which has been done with microbiology. And direct detection of AMR is also being practiced right now. We'll handle this one by one. So what's being proven is a Chilean study which was done in 2020, right in the middle of the pandemic. So what happened was those scientists decided that RT-PCR is taking too long. We want to rapidly identify, isolate, and carry out further epidemiological measures of patients of SARS-CoV-2. Now the challenge they faced was that it did not have those databases or those spectra which are required to identify the organisms. So what they did was they took hundreds and hundreds of known positive and known negative samples, and they prepared a library of their own. That is, they prepared a database of their own, and then running those samples directly from the nasal swab onto the multi-dose site, they compared those spectra which were obtained to those libraries that they generated themselves. So, detection of SARS-CoV-2 in patients' nasopharyngeal swabs was reduced from hours to minutes. So that was one very big advantage of quantitative in those situations. Next thing, probably the most important aspect of multi-toughness and its application to actual patient management was identification of identification of microorganisms from positive blood cultures. So these positive blood cultures which are flashed positive, they are not directly going to be put on the multi-dose machine of the slide. It may not get detected. They might be false negative. So what they did was, they did two things. First was saponification, next is centrifugation. So they did those particular things in order to obtain something known as a microbial pellet. And this pellet was considered to be the actual pure culture and then loaded onto the microbiology machine. So what happened was, the moment these blood culture bottles flashed as positive, immediately they were saponified, centrifuged and tested on monitor. And the identification was obtained in minutes rather than overnight on the next day. So how it helped was, they were obtaining the identification of the organism in the blood culture it will help the clinicians to change, to de-escalate, and actually stop the use of empirical antibiotics. So empirical antibiotics in blood cultures, as we all know, are usually higher antibiotics which are used. So de-escalation can be initiated early. If required, the antibiotics can be stopped early. So this paves the way towards actually reducing AMR in hospital settings. So this is a very, very, very important part in which Microbiology can help the clinician. Now, another thing which is being done right now, not at a grand scale of course, but it is being done right now, is actually intersectoral coordination between microbiologists and engineers, or software engineers to be specific. So what they did was, in Taiwan, what they did was they developed a library of various vancomycin resistant enterococci. They ran vancomycin resistant enterococcus strains and prepared a library just like the one in SARS-CoV-2. So they prepared a library and they taught a machine program with the help of a convolutional neural network to recognize those patterns in normal or clinical isolates. So the multi-top machine was actually only able to identify whether the actual organism was enterococcus or not. But feeding those data and those spectra into the machine learning software with the help of the CNN, it was actually able to identify those particular strains of enterococci which were vancomycin resistant. So direct detection of resistance from the isolate without application of any further tests, whether it is phenotypic or otherwise. This is actually beneficial to the patient, not just to the patient, but also from an epidemiological point of view. That is, application of proper infection prevention and control practices were rapidly done, prevention of an outbreak, prevention of spread of those infections, and of course, reduction in the hospital costs, reduction in stay in the hospital. All those secondary benefits were also obtained from it. So these three important things which are being done with the help of monitor, which are separate from actual microbial identification, whether it be bacteria, fungi, or microbacteria. This is the application which is being done in clinical practice or in laboratory practice these days. So this is quite promising. So can we do more? Answer is of course yes. We can actually increase our collaborations with different kinds of fields, for example in biotechnology, with machine learning, with AI scientists and all, to develop better and better algorithms, better and better machine learning patterns. Because MARDITOF is actually a very, very powerful tool. It can detect any kind of proteins. It's what we do with that understanding of proteins is what matters. So, what has been proposed, what has been tried over the world over? So, since the study about vancomycin resistant enterococci was quite successful, other studies have also been done in the respect of gram-negative bacilli also. This one study from Switzerland, they tried to generate such kinds of databases about the resistance patterns in gram-negative bacilli also, like for example Pseudomonas acinetobacter and so forth, which are more commonly found in hospital settings. So, this particular study or this particular approach is currently not that much in vogue. The reason for that is one kind of genotype or one kind of resistance gene can manifest in many other patterns. So encompassing all those patterns, all those phenotypes and developing spectra out of them is quite a challenging task. And it requires much more application of machine learning and AI development, AI software. So that's why it's something in progress right now, but it shows quite a bit of progress. Now, another thing which is a bit more further down the road in cancer is in cancer detections. So there are two important technologies which I want to discuss over here. The first one is something known as multi-high-plex. So multi-high-plex is technology that they are combining with immunohistochemistry for detection of anaplastic or metaplastic cells from FNAC samples, that is fine needle aspiration cytology samples, which are liquid in nature. So from liquid samples, they are able to generate a pixel by pixel image, that is a dot which represents a cell. So one cell in one particular color is going to signify a normal cell. Cell in a different color, a dot of different color will signify an anaplastic cell, or some or the other kind of mutation is there. So this can give a direct pictorial representation of what is actually happening inside that sample, which can actually help us or help the clinician to determine whether the patient requires further follow-up about cancers or not. So what it does, this kind of technology, it reduces the time for detection of such anaplastic or mutated cells by 4 times as compared to immunohistochemistry. So the crux of the whole matter, the crux of everything which has to do with multi-top is actually saving of time, right? So the next technology I'd like to discuss here is something known as multi-MSI. So MSI stands for mass spectrometry imaging. So the previous technology, it could generate pixelated versions of tissue samples or rather liquid samples. So this technology is going to help us generate such images, actual pictorial or actual images of the tissue which is there, or the proteins of the tissue which is there, from actual biopsy samples. As you can see in the image over here, it is a multi-MSI image of topoisomerase 2 inhibitor in a colon cancer in a mouse model. So these highlighted regions in purple color, they are actually the anaplastic cells which are supposed to be found in the colorectal cancer. So this was done in mouse models. Human testing has not been done yet, so we don't know whether it will work or not. So whether or not it has if developed successfully enough, if proper machine learning integration has been done, would it be able to replace histopathological examination? It's something which we could ponder. Because histopathological examination requires so much of expertise, so much of time, so much of reagents and all that. This is quite simpler in comparison. Right, so now detection of cancers is dependent on various types of omics. Like for example, starting with genomics, which actually has to do with the nuclear or the genes of the person, of the cell, followed by transcriptomics, proteomics, metabolomics, and all those. So all these together, combined together, they are going to form certain biomarkers which are going to ultimately help us in diagnosis of cancers. So multi-top actually deals with the proteomic biomarkers. So basically, what I want to emphasize upon is that multi-top detects proteins. Multi-top is fast. Multi-top is useful in detection of AMR, in surveillance of AMR, and taking actual evidence-based steps in epidemiological prevention of such a spread of such genes. And also it has a lot of strong future in cancer diagnostics. So I'd like all of you to just think over this. Since the topic of the theme of this conference is microscopes to genomics. So talking about genomics, we can say that genes which encode for certain characteristics, they may or may not get expressed. We don't know what is the dominant, what is the recessive. But proteins which are there in the cells, they are actually the expressed genes. Now since multi detects proteins, what do you want to believe? Do you want to believe what can be expressed or not expressed, or what has actually been expressed? I'd like you to think over this. Thank you.
Thank you, Dr. Akshay. First of all, for sticking to the time and for sharing the future applications of the multi-top diagnostic microbiology. Dr. Akshay Karyakartar has some big shoes to fill, and we know he will be up to the challenge. Thank you so much. Felicitate all our speakers at the end of the session. After this, I call upon Dr. Rashmika Das. She is the next speaker. Since Rashmika Das has joined the region is a dedicated clinical microbiologist with over 5 years of experience in research and diagnostics. After earning her master's degree in microbiology from Grant Medical College and Sir J. J. Mulko Hospitals, Mumbai in 2019, she has been associated with Department of Microbiology at DGGMC Pune. Dr. Das has played pivotal role in National COVID-19 RT-PCR laboratory and has extensive expertise in next generation sequencing, having sequenced thousands of SARS-CoV-2 samples under the leadership of Dr. Rajesh Karyakarte. She was instrumental in establishing the Center of Excellence for Genomics at BJGMC in 2022. She is actively involved in genomic surveillance as a part of INSACOG and collaborates with several prestigious institutions. Dr. Das is committed to advancing the role of genomics in understanding and diagnosing the infectious diseases with expertise spanning molecular diagnostics and genomic research. Now I hand it over to Dr. Ashwin Das.
Thank you, madam. A very good morning to everyone. It's an honor to be here today to talk about a topic that is reshaping the landscape of healthcare and public health, that is genomics. In the next few minutes, I'll be exploring the multifaceted role of genomics in transforming medicine. I'll begin by discussing how genomics has revolutionized diagnostics. It will strengthen the pandemic preparedness, driving innovations in vaccine development and is pioneering in therapeutic breakthroughs. So to start with, my first heading would be pathogen identification and discovery in the age of next-gen sequencing. The traditional diagnostic methods often require multiple tests that are hypothesis-driven. On the other hand, with metagenomic next-generation sequencing, we can now detect all microbial DNA or RNA in a sample in a single run. This hypothesis-free approach has been pivotal in identifying co-infections, novel pathogens, and has been helpful in understanding the full microbial landscape. One of the most compelling examples of metagenomics utility was the discovery of SARS-CoV-2 virus in Wuhan in 2019. This breakthrough was instrumental in determining the cause of the mysterious pneumonia outbreak in late 2019, highlighting the technology's critical role in outbreak response and public health. But SARS-CoV-2 is just one example. What about other unknown threats that genomics can help us uncover? So studies have suggested that over 3 lakh mammalian viruses remain undiscovered, many of which pose zoonotic threats. By integrating genomics with machine learning, scientists have already uncovered 1,60,000 new RNA viruses, some from extreme ecological niches. The ability to identify these threats early is crucial for preventing large-scale outbreaks. Now again, also historically, bacteria were classified based on their morphology, growth requirements, and pathogenic potential. And this approach was based on DNA homology of at least 70%. While this served as a foundation, it often lacked the resolution to capture the true diversity within the bacterial species. So with the advancements in genetic analysis, particularly with whole genome sequencing, this has revolutionized the field of microbiology. Researchers now use metrics like whole genome average nucleotide identity or ANI to compare and delineate bacterial species with much greater precision. So this has led to a re-evaluation of established taxonomic relationships. I'll take a minute to give an example here. Looking back at India's first wave of SARS-CoV-2 pandemic, where various variants dominated different regions with a notable shift in variants by April 2020. The wave peaked in September 2020 and began to decline thereafter. However, the country noticed a resurgence of cases in January 2021, predominantly in 3 states, that is Maharashtra, Punjab, and Chhattisgarh. In Maharashtra, apart from the major cities like Mumbai and Pune, a significant rise in cases were seen in districts of Vidarbha, in particular Amravati. The emergence of new global variants around the same period was concerning as they were suspected to be responsible for fresh surge of new cases. On sequencing these samples from Amravati, it was found that Amravati was dominated by clade 21b, which carried immune escape mutations. Due to the presence of these immune escape mutations, the WHO on 4th April 2021 declared Kappa variant as a variant of interest. This variant rapidly evolved to what is now known as Delta variant and was responsible for driving India's second wave. Therefore, when a continuous surveillance system is implemented, it ensures timely and effective public health responses to such emerging variants. It is not just about identifying the variants, the key lies in establishing their clinical relevance. By correlating genetic findings with real-world clinical outcomes, we generate actionable data that can guide targeted public health interventions. This integrated approach is crucial for shaping effective policies, ensuring resource allocation that aligns with emerging threats, and ultimately improving population health outcomes. Pandemics are exacerbated by urbanization, climate change, and zoonotic spillovers. The recent surveillance initiatives by various organizations worldwide now prioritize high-risk viral families. Now, having discussed the global challenges and initiatives to tackle pandemic risks, the next crucial question is how can we ensure timely responses to these threats? This brings us to the importance of early detection systems. Early detection system is critical in preventing and controlling outbreaks, particularly in low and middle income countries that face a higher risk of zoonotic spillover due to limited resources for clinical surveillance. One promising approach here is what is known as wastewater surveillance. Historically, this tool has been highly effective in monitoring poliovirus and responding to outbreaks. And more recently, it has demonstrated its utility with SARS-CoV-2, influenza, RSV, measles, monkeypox, and Zika. When rates of clinical testing are low, wastewater surveillance provides a more accurate picture of the actual burden of the disease in a community. By detecting pathogens shed into the environment, it bypasses the need for individual testing, identifying both symptomatic and asymptomatic cases. This capability makes wastewater surveillance particularly valuable in under-resourced settings or during large-scale outbreaks. It can detect pathogens irrespective of their transmission route and correlate findings with disease incidence in the population. Now, by integrating such strategies into our existing surveillance frameworks, we can bridge gaps in traditional monitoring systems and significantly enhance our ability to respond to emerging health threats. Now, here are some real-time examples from Pune, where SARS-CoV-2 variants like JN.1 and KP.2 were detected in wastewater 3 months and 1 month respectively before the rise of clinical cases in the community. So these findings highlight the effectiveness of wastewater surveillance as a reliable early warning system for outbreak detection and response. Now, the next logical question is how can we expand such approaches to prepare for an inevitable next pandemic? We live in a world where emerging health threats dominate the headlines, be it Mpox virus, rising cases of bird flu, or fears of Marburg virus outbreaks. The critical question is not if the next pandemic will happen, but when. So how do we prepare for it? One promising solution is investing in wastewater surveillance, which offers numerous benefits, that is preparedness, scalability and cost effectiveness, and supports data-driven public health measures. So as we brace for future health crises, incorporating wastewater surveillance into our public health frameworks is not just an option, it's a necessity to stay ahead of emerging threats. is antimicrobial resistance, as we call it as the silent pandemic. It poses a significant threat to global health. Misuse of antimicrobials continue to drive the evolution of resistance, calling for innovative solutions, improved surveillance, and public health education. Genomics is at the forefront of addressing this crisis through several key applications, either by augmenting the phenotypic testing that we do or understanding the gene diversity for better characterization of these genes and their variations. It helps in detection of common antibiotic resistance genes and at the same time addresses the challenges of undetected genes, thereby filling the gap by uncovering the hidden resistance mechanisms. The studying AMR evolution in pathogens is also important. Cases once thought to involve new strains often reveal evolutionary adaptations of existing strains, reshaping our understanding of infection dynamics. AMR is not just a human health issue, but a One Health challenge affecting humans, animals, and the environment. Genomics helps to track the transmission of AMR genes between humans, animals, and environments, enabling targeted interventions. By integrating genomic approaches into One Health strategy, we can better understand the interconnected drivers of AMR and develop comprehensive solutions to address this multifaceted challenge. Now let us shift our focus to the healthcare settings. Healthcare-associated infections remain a persistent challenge in healthcare settings despite the implementation of infection control strategies and bundles. Now, traditional microbiological methods often fall short when it comes to determining the true relatedness of organisms, which is crucial for understanding the spread of infections. Now, this is where NGS plays a transformative role. It allows us to pinpoint the exact source of an outbreak. It provides detailed insights into how pathogens are transmitted between patients, between healthcare workers, and even between healthcare facilities. NGS offers a comprehensive view of how antimicrobial resistance genes evolve and spread within and across the healthcare settings. Now, I'd like to give a very interesting example here. Building on the role of genomics in infection control, we conducted a study at BJ Government Medical College in collaboration with Johns Hopkins University. In this study, what we did is we sequenced 11 E. coli and 15 Klebsiella pneumoniae carbapenemase resistant strains, and we wanted to see what were the resistant genes present. As expected, we detected the presence of carbapenemase resistant genes, various carbapenemase resistant genes. However, what stood out were additional findings beyond antibiotic resistance, and we also detected what is called today as disinfectant resistant genes in these isolates. So this discovery has significant implications for each and every infection control practitioner, as these genes can potentially reduce the efficacy of routine disinfection protocols in healthcare settings. The detection of disinfectant genes in carbapenemase resistant isolates not only highlights a new dimension of antimicrobial resistance, but also raises a critical implication for hospital infection control. This can lead to persistent contamination of hospital surfaces and equipment, may increase the risk of healthcare-associated infections, particularly in high-risk areas like intensive care units where patient vulnerability is higher. There can be an increased need for enhanced surveillance of disinfectant-resistant genes, where hospitals should routinely test the efficacy of disinfectants being used to ensure that they remain effective against the evolving pathogens. Additionally, they may also need to explore alternative disinfectants and advanced cleaning strategies to mitigate these risks. Now, another exciting area where NGS has been leveraged is paving the way for innovative diagnostic kit development. It can identify the entire genome of the pathogen and guide the design of region-specific diagnostic tests based on circulating strains. It will also enable the creation of multi-pathogen panels to test for multiple diseases simultaneously, ensuring that the diagnostics stay relevant for rapidly evolving diseases. It will help in generate a genomic database to build pathogen databases, aiding future diagnostic development and standardization. I'd like to share a very interesting example here. Back during the Omicron outbreak, WHO came out with a regulation where they said for countries using diagnostic kits that include S-gene target failure or SNP detection assays to screen the variants, those samples should be recommended for sequencing and to characterize the circulating SARS-CoV-2 variants. And they said whenever available, these genomic sequences and the S-gene target failure data should be shared with the WHO. Now what we did during this period, we sequenced all the samples that were available and to check if these S-gene target failures were actually Omicron variants. We saw a very interesting graph. The blue ones show the blue lines show the isolates which showed S-gene target failure, and the orange ones which were non-S-gene target failure samples. So you see a very nice wave there. Towards the end, as we know, the Omicron, towards the end of December, the Omicron cases were rising. So what we saw, so what should we expect? That the S-gene target failure should be increasing. But what we saw was completely opposite. S-gene target failure cases started decreasing. So did that mean that Omicron cases started falling? No. It was the Omicron had evolved into multiple lineages, that is BA.1 and BA.2. BA.1 showed S-gene target failure, whereas BA.2 did not show S-gene target failure. And here we missed detecting BA.2 if you would have used that particular kit. So here it guides us whether to continue to use a particular kit or to discontinue it. So it helps in policy making. Now with the ever-evolving nature of pathogens, traditional method often falls short to address the emerging challenges. So it offers a groundbreaking approach to revolutionize how we design our vaccines. So vaccines have always been a cornerstone of public health, but methods used to develop them have evolved significantly over time. Now, NGS has allowed the researchers to predict the antigenic proteins exposed on the surface of the pathogen, and using reverse vaccinology approach, these predicted antigens can then be experimentally tested for their ability to elicit immune response. The most remarkable outcomes of NGS-driven vaccine development is the rapid creation of mRNA vaccines for SARS-CoV-2. Now, why this matters is with NGS, we can address the challenges like antigenic variability and emerging pathogens in real time. This is especially critical in responding to outbreaks where rapid vaccine development is essential. It also enables the design of vaccines tailored to specific strains circulating in different regions, increasing their effectiveness. Now again, I'd like to share a study from IISc Bangalore, where the study highlights the significant mismatch between currently available dengue vaccines and the dengue strains circulating in India. It shows antigenic divergence where the prevalent Indian dengue virus strains differ significantly from those used in the tetravalent vaccines, which are based on the oldest strains isolated from 1964 and 1988. Following this study, researchers developed a novel DNA vaccine candidate combining the envelope protein domain 3 of dengue virus serotype 1 to 4 and non-structural protein 1 of dengue virus 2. Now the study emphasizes the utility of the consensus genus approach in conjunction with DNA delivery for the development of vaccines against various emerging and re-emerging viruses. Now coming to another transformative application of genomics, that is leveraging the human microbiome for therapeutic development. The technology has revolutionized our understanding of human microbiome, an intricate community of bacteria, viruses, fungi, and archaea residing in and on our bodies. The microbiome is now recognized as a key player in health and disease. We know very common examples like recurrent Clostridium difficile infection, where fecal microbiome transplant is already in use, and there are several clinical studies which are undergoing to investigate the use of vaginal microbiome transplantation to treat symptomatic and intractable bacterial vaginosis and in HPV infected women. Another critical and the last application of NGS that I am going to discuss is the transformative impact on clinical practice, precision medicine, particularly in oncology. It allows the discovery of tumor-specific mutations, enabling personalized treatment, plans tailored to genetic profile of an individual's cancer. By detecting mutations associated with hereditary cancer syndromes, NGS supports early risk assessment and preventive strategies. It also aids in selecting targeted therapies by identifying actionable mutations. Now, this is a very powerful example of a next generation sequencing which is transforming the clinical practice in real time. Using NGS combined with machine learning, surgeons could achieve enhanced diagnostic accuracy for central nervous system tumors even during surgery. So within 90 minutes during the live surgery, they could take a decision whether to resect a brain tumor or not to resect a brain tumor. So that is a real-time application of NGS where surgeons could take a decision within 90 minutes to treat a brain cancer. Now another example, and the last example, perhaps which illustrates how genomics can play a life-saving role in chemotherapy safety and precision medicine. Certain drugs such as fluorouracil and capecitabine can cause severe and even fatal toxicity in patients carrying genetic variants in the DPYD gene. Now the statistics are striking. 2 to 3% of patients carrying one DPYD variant can experience fatal toxicity. Now this highlights a critical need for pre-treatment DYPD genetic testing to identify at-risk individuals. Now genomics not only helps prevent adverse outcomes but also ensures the patients receive safer and more personalized therapies, revolutionizing the cancer cure. To summarize, genomics is revolutionizing healthcare by transforming diagnostics, enabling rapid and accurate pathogen identification while guiding pandemic preparedness through tools like wastewater surveillance to track emerging variants and zoonotic diseases. It has redefined vaccine development, addressing antigenic diversity to create region-specific and effective vaccines and advanced precision medicine by tailoring treatments to individual genetic profiles for improved outcomes. Together, these advancements highlight the immense potential of genomics to tackle global health challenges and improve patient health care. Thank you.
Thank you, Dr. Rashmita, for precisely telling us the role of genomics in various diagnostic and research fields of microbiology. Thank you so much. It gives us a proud feeling when a new generation is delivering a talk is our own student. I call upon Dr. Madhuri Somani. Madhuri Somani is a University Gold Medalist from BJ Medical College, Pune. She is currently working as Consultant Microbiologist and Infection Control Officer at Pushpavati Singhania Hospital and Research Institute at New Delhi. She is the Secretary of Hospital Infection Control Committee and the ANSP. Dr. Madhuri has recognized for proficiency in leading and managing the high-quality microbiology laboratories, particularly in tuberculosis diagnostic. She has played a vital role in implementation and maintenance of hospital infection control programs credited for zero non-compliances. She is a faculty member for Outbreak Investigation Workshop, sharing knowledge and skills in managing infectious diseases outbreak effectively. She has authored multiple scientific papers and contributed to both chapters on diagnostic microbiology, infection control, and biomedical research. She is a mentor and guide for clinical DNB residents, fostering the next generation of healthcare professionals. I request Dr. Madhuri to deliver a talk.
Good morning. Thank you so much, ma'am, for that kind introduction, and I take this opportunity to express my gratitude to the organizers to give me an opportunity to speak today. So, with whatever little time is left, ma'am, I try my best to stick to the time schedule. Keeping in sync with the futuristic theme of this conference, I shall be speaking upon total lab automation, the road ahead. So I'll be delving in the next 15 odd minutes on the why and what of total lab automation, the initial impediments and drivers towards total lab automation, what are the current systems available and their benefits, and with them come challenges and future directions as well. So, in a technical definition, what exactly is automation? Automation is essentially a technology by a procedure that is performed without human assistance and intervention, and in contextual laboratory, it's essentially switching from manual, repetitive, moderate value tasks to machine-based tasks. And I think our clinical biochemistry and hematology counterparts are already doing their entire workload on the 6-mexes and ABOs, and about 80 to 90% of their work is already automated. So automation is relatively late to hit our field. It's only been 30 years since the first automation systems, starting with the blood culture systems, actually came into the market. Following that came the ideal susceptibility testing equipments as well. And those of us who have been using Bactex, Bacteroids, Vitex and Phoenixes know how well this partial automation has actually changed the way we work and how much it has contributed to the productivity of a clinical microbiologist's work. So if partial automation can add so much value, just imagine what total lab automation could do. So let me just be clear about this, that what we're talking about here is the automation of the steps of the clinical bacteriology diagnostic workflow that would start from inoculation to the final reporting. And this particular entity, TLA, has only recently been recognized as a valuable component of our laboratory. They had their own impediments. What exactly obstructed? Why was automation TLA late to come to clinical microbiology? So unlike our other clinical lab medicine counterparts, microbiology is often being considered too complex to automate. We get all sorts of highly heterogeneous samples in addition to our urines and stools. We get tissues, we get prosthetic devices, we get catheters in all sorts of sizes and shapes of containers. So it's so difficult to, process such myriad kinds of samples. We have so many culture media that we have to inoculate along with the enrichment rods. We have streaky, quantitative, semi-quantitative. So this has been an impediment. Then next comes the cost of automation. Unlike the huge chunk of workload that goes to hemat or biochemistry, the cost of it has always been thought that microbiology laboratory investing in TLA wouldn't really be cost effective, which I think I'll speak about in my subsequent slides. Microbiology laboratories have traditionally been considered too small for automation, which I believe is still a fact in our country. And can any machine ever replace the critical thinking skills of a microbiologist and a medical lab technologist? The study and research says otherwise. So, what exactly has been the driving force behind the total lab automation in our laboratory? Why this shift of paradigm? So the most important shift has been because of the increasing quality requirements. With the patient-centric approaches now, we focus more on lowering our TAT and increasing traceability of the samples because it's an important, essential part of our diagnostic stewardship workflow. So this has been the biggest driver worldwide. Liquid-based transport and elution medium is available now. But we have these LVM devices into which samples can be directly eluted and loaded onto the TLA, so that is standardized sample processing. What about the industry changes? I think this particular point applies more to the Western world than our country, who are suffering from personnel shortages, and they'd like to cut down their workload time and make everything more technical. Their labor force has been static for the past 10 years as per this data, so they want to shift everything over to automation. MALDI has been a game changer. Dr. Akshay was very eloquent about how rapid, accurate MALDI can be, and most importantly, MALDI is amenable to automation. AI and machine learning, there was this brilliant symposium yesterday. We're looking into the future with all our chat GPTs and additional AI software. So, TLA is here. So So what is the current landscape? In the next few slides I'll be talking about the systems currently available, and briefly, because of paucity of time, we just touch upon the various technical features. So currently there are just two systems, one from Copan and the other from BD, that have actually seen some commercial success with and have been quite established in various diagnostic laboratories throughout the world. They are primarily these multifunction modular stations which cover all the steps of the workflow of the laboratory. So to be able to I just simplify the complicated modular system into, I'm breaking down into the inoculation systems which do the sample barcoding and the automatic streaking, followed by the smart incubators, which are equipped with the cameras which give you HD images and automated colony picker and disk dispenser for automatic susceptibility testing. And these modules are connected to each other with conveyor belts which are unidirectional or bidirectional. So this is in a work from BD Kiestra, in which there are various modules which sorts the media, identify, barcode it and inoculate it, with the conveyor belts that take it to the normal and carbon dioxide incubators. And these conveyor belts also deliver culture plates to the workbenches for follow-up. Similarly, for WASP, unlike three separate modules, their inoculation system is a single module which can store, barcode as well as do inoculation. And there's a one-sided transport of the plates which delivers it to the normal and carbon dioxide incubator and later deliver it to the output stacks and the workbenches as well. So this picture from a study where TLA has been used for the past 4 years shows that about 97% of samples they claim are automatable. Only the rest 3 samples like tissues and catheters and devices, which constitute less than 3% of the total lab workload, are actually still needing manual processing. So I'll just briefly touch upon the technical aspects of the inoculation systems. So I won't be describing this in details due to the paucity of time, but there is a slight difference between the inoculation system, the way the streaking and inoculation is done in Kiestra and Copan. In Kiestra, it's a bead-based inoculation in which the streaking is done in the closed culture plates and it minimizes the aerosolization of the sample. But in WASP, they have a robotic arm in which sample is delivered by a loop and is streaked with a loop and a spreader of the sample. Next are the smart incubators, which are equipped with the HD cameras. Now these incubators, because ensure automatic delivery of the culture plates, the samples that are barcoded, and it's ensured that these particular culture plates are in a unique location within the incubator, and because they've been located in a unique location due to the correct barcoding, it's very easy to trace these culture plates. It's very easy to call them, take their digital images and basically do a follow-up of the plates. And the constant and uniform laminar flow is also ensured because there is a decrease in time to inoculation and transport of culture plates to the incubator. I will not go into the details of the technical aspects of both incubators because of the paucity of the time. But roughly, they have a robotic arm which move the samples up and down and around the incubator. They either have a static or they have a mobile carousel. Like I mentioned, they are equipped with the HD cameras, which enable extremely high definition images under various illumination conditions to be able to study the morphology of the colonies very well. They are equipped with the time-based visualization in which you can continuously track either at 2 hours, 4 hours, 8 hours, and so on. So you can keep a track of the growth of the colonies. The automated colony picker and disk dispenser. After the high resolution images of plates at specific intervals, the technologist would then pick which colony to follow up. So the automated colony picking in some culturing, if required, can be done by Colibri. Now this case, BD still doesn't offer automated colony picker and disk diffusion dispenser. They are in the process of working on it. But Colibri is currently available in the market, which can pick colonies with high efficiency and with very high accuracy can make clear spots on the multi-plates to enable early recognition. And Radian allows us to put up AST as per the UCAST guidelines, there is an automated dispenser and software protocol enabled to do it. So with all this technology, how exactly has it improved the productivity and efficiency of the laboratory? There was plenty of literature that I had to analyze, which highlighted that they have improved the standardization, they have increased the efficiency and quality, but of course their workers safe with their cost benefits, and they've had an impact on patient care as well. For instance, this particular study showed how, on comparison between the automated inoculator and manual urine specimens, the isolation of the colonies from them were significantly more. We had a higher number of detection of different morphologies of bacteria, and manual and wasp lab inoculator had an excellent agreement when it came to the CFU per mm as well. Similarly, there was this study that compared pre and post inocula, the PTK stress inoculator and the urine specimens, and it was observed there was a 3 to 10 fold of higher leukodiscrete colonies. The technologists claim that it was easier to read these plates due to consistent streaking patterns. There is no inter-technologist variation, and of course that came down significantly. TNAs also improved the isolation of the fastidious pathogens. For instance, this study was very interesting. Not only did it tremendously increase our normal uropathogens isolation, but there was much higher isolation of gram-positive bacteria, and there was a whopping 371% increase in Neisseria gonorrhea. I cannot imagine isolating a Neisseria gonorrhea so easily on a culture plate. Similarly, this particular study, pre and post TLA on urine culture, significantly showed higher isolation of hitherto unknown probable uropathogens like actinomyces and actinodectignum. Pre and post TLA, there were comparable amounts of GNV along with significantly more of these unusual bacteria. The TAT was lower significantly, and this study also highlighted that these automated inoculation enhanced growth of non-classical and rare culture bacteria from the urine sample. So you never know, these bugs might eventually make it to the list of the uropathogens. It has tremendously improved the lab efficiency. This study, pre and post TLA done on urine swabs and e-swabs, showed a decrease in a median TAT, very significant drop from 52 hours to 22 hours, and decrease in TAT reporting of MRSA, VRE, and ESPOs. Similarly, another study on urine, which was done along with Maldi, showed a decrease in time to identification, decreased in time to EST results, as well as a statistically significant decrease in time for negative cultures. This particular study was done on blood cultures, which showed a significant reduction of time to results with an 8-hour incubation. Traceability, of course, it's a very important parameter of the quality because there is a minimal human intervention, because the samples are so accurately barcoded. It's so easy for you to actually real-time monitor where your plate is going. And at any point of time, it's amenable to do a comprehensive audit of the entire diagnostic work. So the studies have been done also on cost analysis. This particular study showed savings of about $100,000 when they analyzed it pre and post total lab automation. This recent study in 24 actually was the biggest multi-centric study that was done in the USA, and the cost avoidance in this particular study was calculated, in which these 4 labs, after implementation of TLA, saved thousands to millions of dollars on their labor. There have been few studies on patient care, but whatever few have been done, they've shown a significant reduction in duration of empirical therapy. So TLA does have an impact on antimicrobial stewardship outcomes as well. Increased workplace safety, needless to say, minimum handling, minimum exposure to the samples and the culture plates. So it definitely enhances worker safety. But there are still future directions and research needs which is still in works in making the remaining steps manual, automated, I'm sorry. For instance, Colibri, apparently it's a well-established system, but there are still a lot of validation studies going on with Colibri that is being offered by Wakad, in which Colibri versus manual spotting with Vital MS was done, and the agreement was excellent. It was about 99.5%. Similarly, Radiant for putting on the antimicrobial susceptibility testing was compared with the manual test diffusion, and the categorical agreement was acceptable. More studies are being done particular study evaluated rust of from the blood culture model with the complete TLA using colloquial radial and then showed categorical treatment. Radial is still being studied in various validation studies. I won't delve into how impactful AI has been in growth monitoring and image analysis. There was such an excellent, such a brilliant talk on AI in the symposium yesterday. So with all these benefits come a few challenges as well. I'd like to just emphasize upon the LMIC perspective because we still, we would want to assess if TLA would fit in the landscape of our country. There is high initial and operational cost, there is a lot of capital cost involved, and needless to say, we still suffer from infrastructure issues. What about the small and medium-sized labs? Can they incorporate TLA even if you invest in it? And TLA, even if it comes, would be more oriented to the urban areas where we have high throughput labs. And workforce will be need to train accordingly, like Dr. Balaji mentioned yesterday. The training would be, we would need to be trained with microbiology of bioinformatics, microbiology of data sciences. So the way we learn microbiology is going to change tremendously. So having said that, TLA adaptability in LMIC still needs to be studied well. Publications have been from the West. So impact on personnel. When I first started reviewing literature for this, the first question that came to my mind was, okay, what's going to happen to my job, the job of the lab technicians? So there is a concern, yes, there will be a shift of job role from the biosafety cabinets and your workbenches to basically in front of the computers where you sit and analyze the digital images and you fish out data. So your working is going to change. Now will there be loss of jobs? As compared to the West, we actually have a growing young workforce, more qualified workforce. So it's hard to say there will be loss of jobs, but unless we train ourselves well, if we align ourselves with the needs of practicing microbiology in a futuristic way, it might not happen. And those of us who still love the conventional way of working worry that we might lose what we've learned. It was so hard for us to learn the characteristics of colony, the consistency, the size, shape, and all. Are we going to forget it? But, like I said, upskilling would become very important. And, once your workflows, optimize after all that hard work, it's going to be satisfying at the end of the day. Because all your strain of doing this mundane tasks, it would become automated. There are some equipment-related challenges that TLA users have published that, sometimes contamination with the specimen processors and incubators do happen because you really don't know what your sample has, right? Maintenance is very expensive, and what do you do when there is a breakdown and when there is a downtime? You have to go back to manually processing everything again. And software does come with its set of challenges. There will be bugs, there will be software-related issues. And currently there are no integrated incubators for anaerobic cultures, microaerophilic cultures in the TLA systems. They're only offering carbon dioxide and novel incubators. So I'd like to quickly wrap up and summarize what I spoke about. TLA is going to bring about a transformative shift for clinical microbiology. It's going to significantly alter our workflow once we start using it. Currently, there are just 2 commercial systems that have been extensively studied and they're still being rigorously evaluated. They come with their own set of numerous benefits like increased efficiency, increased workplace safety, excellent quality parameters. They do have the challenges of high capital costs, resource limitations, especially in a country like India where we still struggle from a constant power supply and a consistent IT support. But advances in AI, connectivity and integration would lead to a faster, more accurate and personal diagnostics, and I'm sure it will improve patient outcomes and public health outcomes eventually. So thank you so much for that patient listening.
Thank you, Dr. Balaji. We all know that the microbiology laboratories are incomplete without automated machines. So, to conclude, the diagnostic microbiology is always inspiring, and we hope that this new generation, the budding microbiologists, will contribute significantly to our field of microbiology. Thank you so much. I request all the speakers to come on the dais.
अरे सर तो मारना पड़ेगा मैं करता